This lecture continues our series in genomics and diversity. Today, we'll do the exercise in genome analysis that we referred to in the last lecture. By the end of this lecture, you should be able to use IMG to analyze a genome. You should be able to find genomes of rumen and other bacteria, determine what metabolic pathways a genome encodes, and make inferences for the metabolic capabilities of bacteria. In the exercise today, you will analyze the genome of Streptococcus bovis, or Aquinas, JB1. In this analysis, you'll make a simple prediction: if it can ferment mannose to lactate and butyrate. So the central question we're trying to ask today is, can Strep bovis JB1 ferment mannose to lactate and butyrate? We can break down this question and ask, can it transport mannose? Can it form pyruvate from mannose? Can it form lactate from pyruvate? And can it form butyrate from pyruvate? Breaking down the question in this way will make it easier to address during genomic analysis. Before we can answer this question, we first need to access IMG and find the genome for Strep bovis. To access IMG, we go to img.jgi.doe.gov, as shown here. We scroll down, and we click to enter the main website. Once we're at the main site, we can find the genome for Strep bovis JB1. We go to Find Genomes and Genome Search. Then type in Streptococcus equinus JB1 and click Go. To use the genome, we have to add it to the genome cart. We have two genomes of Strep bovis JB1. We'll pick the first one. Hit Select, and click Add Selected to Genome Cart. We can now analyze the genome. We'll access the KEGG pathways by going to Find Functions, KEGG, and Pathway via KO terms. We're led to a screen that shows us all the KEGG pathways. This is what we'll use to do our analysis. To determine how we should start our analysis, let's go back to the central question. In order to answer if Strep bovis JB1 can ferment mannose to lactate and butyrate, we have to answer first if it can transport mannose. If it can't transport mannose, then it's doomed from the start. To determine if it can transport mannose, we go to the KEGG pathways. We scroll down, and we find phosphotransferase system PTS under membrane transport. We click it. We click View Map for selected genomes. Select our genome and click Add. We then view the map. Once we have the map, we need to examine it. The map shows several different PTS transporters. We can see the transporter for mannose under MAN family. It's composed of the proteins MANX, MANY, and MANZ. Here and throughout, when a box is purple, that protein is encoded by the genome. You'll note that all three boxes for the mannose PTS are purple, and so the transporter is in fact possessed. Going back to our central question, we can say yes, Strep bovis JB1 does transport mannose. What we need to answer next is, can it form pyruvate from mannose? To answer this, we go back to the KEGG pathways, through Find Functions, KEGG, and Pathway via KO terms. You can also hit the back button. We go to fructose and mannose metabolism. And as before, we click View Map for selected genomes, select our genome, hit Add, and View Map. Using the map that appears, we examine if mannose-6-phosphate can enter glycolysis. We see a single box, which represents mannose-6-phosphate isomerase, connects mannose-6-phosphate and glycolysis. Specifically, it's fructose-6-phosphate that's formed and enters glycolysis. We see that the box for mannose-6-phosphate isomerase is purple, indicating that it's present and mannose-6-phosphate is in fact transformed to fructose-6-phosphate and can enter glycolysis. To continue to the map for glycolysis, we click on the glycolysis icon. We now see the map for glycolysis. We need to check if fructose-6-phosphate, which was the product of the last map, connects to pyruvate. We see several boxes representing glycolytic enzymes that connects fructose-6-phosphate and pyruvate. As we can see, at least one of the boxes at all steps is purple. That means the full pathway is present, and fructose-6-phosphate can in fact be converted to pyruvate. Note that at some of the steps, there are multiple boxes, and not all of them are purple. For example, between fructose-6-phosphate and fructose-1,6-bisphosphate, there are 3 boxes and only one is purple. These different boxes are isoenzymes, and only one has to be present for that pathway step to have activity and for the conversion to be possible. So in sum, fructose-6-phosphate can be converted to pyruvate, and as we saw in the last slide, mannose-6-phosphate can be converted to fructose-6-phosphate. This means that mannose can be converted to pyruvate. Let's go back to the central question. We've now shown that pyruvate can be formed from mannose. The next step is to determine if lactate can be produced from pyruvate. To determine if lactate can be produced from pyruvate, we stay on the same map as we used previously. We can see that there's one box representing L-lactate dehydrogenase that connects pyruvate to lactate. It's purple, which means lactate can in fact be formed from pyruvate. And so when we go back to our central question, we say yes, lactate can be formed from pyruvate. Finally, we need to answer if butyrate, otherwise known as butanoate, can be formed from pyruvate. To do this, we go back to the KEGG pathways, again through find functions, KEGG, pathways via KO terms. We then find butanoate metabolism. KEGG calls it butanoate, not butyrate. And we click it. We go to View Map for selected genomes, select our genome, click Add, and click View Map, just as before. Now that we're at the map for butanoate metabolism, we need to see if pyruvate can be converted to butanoate. We examine the pathway connecting pyruvate and butanoate. And we see that there are several boxes that are white. They represent proteins in the pathway that are not possessed by strep bovis JB1. This means that strep bovis cannot form butyrate from pyruvate. Going back to the central question, we mark that no, strep bovis JB1 cannot form butyrate from pyruvate. So in sum, we can say that strep bovis JB1 can ferment mannose to lactate. It can transport mannose, form pyruvate from mannose, and form lactate from pyruvate. However, it cannot ferment mannose to butyrate because it does not possess the enzymes to convert pyruvate to butyrate. The take-home messages are that genome analysis can predict what are the metabolic capabilities of microbes. Making these predictions is simple and fast, as we've just seen. It can be attractive to rely on them, but ultimately, the predictions need to be verified experimentally.